Thanks to everyone for attending, and I'm happy to walk through the data presented in our poster detailing the anti-COVID internal research campaign we completed last year at Veris. I'll walk through the experimental design over the course of the presentation and how we leveraged our ability for rapid antibody discovery using both single B-cell cloning and accelerated hybridoma workflows. Both strategies start with rapid immunizations followed by primary cell harvest from tissues of interest, and we use two different mouse strains to leverage the advantage of each one. The alloy GK mice provide a wild-type-like immune response, but with the advantage of producing fully human antibody variable regions, while the DiversimAb mice produce murine sequences, but elicit an enhanced immune response to broaden the epitopic diversity and ensure valuable regions of the antigen are effectively targeted. Both strains responded well to the immunization protocols, and detectable serum titers at greater than 50,000-fold dilution were observed. In the single B-cell workflow, plasma cells were loaded onto OptoSelect 14K chips with the beacon, which uses optoelectric positioning to guide single cells into individual nanopens. After cell loading, various capture and detection reagents are imported into the channels above the pens, and antibodies secreted from the plasma cells diffuse into the channel, forming blooms during the time-lapse imaging as they bind to the target reagents. Because the assay reagents are separate from the B cell, we can then flush the reagents from the chip without disturbing the B cell and import a second assay with new reagents to perform sequential assays. Once the B cells of interest are identified, we can export single cells into PCR plates for downstream sequencing. For this particular study, we ran two sequential assays. In the first, we were looking to identify high-affinity candidates to the monomeric S1 domain of the spike protein. By capturing secreted IgG on anti-mouse polystyrene beads at the mouth of the pen, we detected total IgG with an anti-mouse secondary antibody in the FITC channel and positive binding to the S1 protein with directly labeled with Alexaflor 647 in the Cy5 channel, and this was in a multiplexed assay format. Then, after flushing the channel, a second mixture of assay reagents was imported, this time with the S1 protein pre-complexed with a molar excess of ACE2 protein. In this format, a lack of S1 signal indicated competition between the antibody and ACE2 for S1 binding to identify potential blocking candidates. On the hybridoma side, thousands of colonies were screened by ELISA, and clones showing reactivity to the S protein but not to an irrelevant counter-screening reagent were pulled forward for secondary screening confirmation by ELISA, followed by single-point kinetic analysis to the trimeric S protein to determine avid affinity using the octet. Recombinantly expressed antibodies from the single B-cell workflow were also available for screening, so they followed the same triage cascade. And out of the hundreds of binders discovered, dozens of clones were selected for full validation, which included various SPR-based assays on the Cartera LSA. A representative subset of these clones is shown here, and full monomeric affinity to the S1 protein was determined in a titration series. Next, a blocking assay was performed to identify clones with potential neutralizing activity. In this sequential format, the experimental antibody was pre-bound to the S1 protein and then exposed to ACE2 protein. The amount of ACE2 binding was quantified and reported as a % increase in binding, with candidate antibodies capable of blocking the S1 ACE2 interaction showing negligible ACE2 binding, like clone 11955 shown in yellow. For all the Cartera studies, two different formats were used head-to-head to validate the data from each one. In the most simplistic version, purified experimental antibodies were directly coupled to the chip surface prior to introducing analytes, and in this case I'm showing the S1 protein binding to an immobilized experimental antibody before introducing a second candidate antibody in a classical epitope binning experiment. Recently, a second assay format's been validated on the LSA where antibodies in crude supernatant can be used, eliminating the time and cost associated with purification and buffer exchange. The setup is slightly more complex, but we found the results from both formats are very similar, and an extra 1 to 2 weeks could be saved from the overall project timeline using the crude sample format. Analysis of epitope binning data revealed a conserved epitope for the ACE2 blocking region, with some candidates binding on the periphery of this core. Multiple other non-blocking candidates localized to different epitopes on the S1 protein, some adjacent and some distal to the blocking epitope. Looking at community plots as another way to visualize the epitope binning data, we can clearly see 6 distinct epitopes targeted by this subset of top candidates, and can quickly establish which ones localize to the receptor binding domain to select high-value candidates. Then, by combining the in-depth biophysical characterization with detailed sequence analysis, we can select lead candidates from the larger pool. Taking sequence similarity into account to define clonotypes, 3 different unique groups of blocking clones emerge. And for a group with multiple clones, we can incorporate in silico analysis of common sequence liabilities to further downselect. And in this case, clones 21C3 and 11955 have very few predicted liabilities, making them excellent candidates for downstream development without the need for extensive engineering. Interestingly, these two candidates have the same sequence, but were each independently discovered with the hybridoma workflow or single-cell workflow, which validates both platforms for discovering high-value leads. Looking at the timeline, the single B-cell workflow discovered sequences in about 1 month, with full validation just 2 months later. On the hybridoma side, the timelines are roughly doubled, but could still be considered rapid when taking into account the large amount of data collected in just a few months. And I'd like to thank everyone who contributed to the work at Abveris and Cartera, as well as Alloy Therapeutics for providing the mice. Thank you.